So today's class, we are going to look at genes and genetic diseases, as we had agreed last week. It will be just a brief lecture, being a holiday, but I'll try to capture the things that I want you people to know about genetic diseases. The objectives of the class will be: one, I'll highlight some concepts that we mentioned last week that concerns today's lectures, just a small reminder. And then I will go to specifics of today. We'll look at the basic structure of the DNA and also explain the process of DNA replication. We'll define a gene and we'll state the importance of a gene. We will also review the process of protein synthesis because it has some implications on today's class. Then we look at gene mutations and state the mechanisms and outcome of gene mutations. Then we'll classify genetic disorders and give some examples for each category. We look at the interaction between the environment and the genes. And we'll finish with what we call epigenetics. So we'll define what it is, we'll state its mechanisms, and we'll give examples. Let's begin with the first agenda there. Just some background information from what we said last week. So last week we said that a cell consists of three major components: the cell membrane, cytoplasm, and the nucleus. Those are the three basic components of a cell. If we narrow down to this nucleus, the nucleus of a cell is usually covered by a membrane around it, what we call the nuclear membrane or nuclear envelope. This is the nuclear envelope. This nuclear envelope has some holes within it that allow passage of some substances, some particles pass through the nuclear pores. Some of the substances may be coming from the cytoplasm to enter the nucleus, or they could be coming from the nucleus and enter the cytoplasm. Within the nucleus itself, we have a lot of things. For example, we have the nucleoplasm. The nucleoplasm is the fluid within the nucleus. We also have the genetic material within the nucleus. And we have the nucleolus. Remember that the nucleolus is the site of synthesis of ribosomal RNA. Most cells will have just a single nucleus, but there are some cells that lack nucleus, like the red blood cells, doesn't have a nucleus. And some cells have more than one nucleus. For example, cardiac muscle cells, skeletal muscle cells, and some white blood cells, they have more than one nucleus. What are the key functions of the nucleus? It controls cell division as well as other activities within the cell. The site of synthesis of the ribosomal RNA, which in particular takes place within the nucleolus. We can say that the nucleus stores, protect, and control the genetic material. We can also say that the nucleus is responsible for replication and repair of the genetic material. Last but not least, the nucleus is a site of transcription of the information that is stored within the genetic material. It is on account of the last three functions that we want to discuss more today. Let's discuss the nuclear genetic material at greater detail. So the genetic material in the nucleus exists in form of chromatin material. When you talk of chromatin material, we are referring to the DNA together with the proteins that hold it. These proteins are many, but the majority are the ones called histone proteins. There are two types of chromatin materials. There's one that is relatively dispersed. And because it is relatively dispersed, it is transcriptionally active. We call that the euchromatin. So you'll find euchromatin in cells which are actively involved in transcription. The opposite is heterochromatin. Heterochromatin is heavily condensed. These darker ones, heavily condensed. Such condensed chromatin material is not available easily for transcription. So it is transcriptionally inactive. We went ahead also to discuss the levels of organization of the DNA. We said that there are 6 levels of DNA organization. The lowest level of DNA organization is a double-stranded molecule. We will revisit that shortly when we talk about the detailed structure of the DNA. But as an overview, there are 2 strands of DNA side by side. And so the molecule is a double-stranded molecule. This double-stranded molecule is the lowest level of DNA organization, beyond which now there's no DNA. Okay, the double-stranded molecule twists on itself to form the helix structure, which is the second level of organization. So the 2 strands are twisted like a rope, twisted on one another to form the helix structure. The helix structure usually goes around the histone proteins, and usually it goes around twice. The helix structure that has gone around histone protein twice makes what we call the nucleosome. So a nucleosome refers to the DNA helix that has gone around a histone protein twice. Several nucleosomes are compacted together to form the chromatin material. And that's the form within which DNA usually exists in most of the cells in the body. However, if this cell needs to divide, for example, remember that cells need to divide to pass that genetic material to the daughter cells. And the daughter cells also therefore need to have the same amount of genetic material like that of the parent cell. So if it was possible to count the genetic material, for example, and this I'm just taking arbitrary. Let's say that a parent cell has 200 DNAs, and that we are supposed to then, this particular cell need to divide into 2 daughter cells. If this cell is not bright enough, it will divide, but the daughter cells will have 100 DNAs each, because it will split its DNA into 2. If you are to think of that being true, it would mean that after some generations, we will definitely not have DNA. So with that in mind, you should imagine what you would be if you trace yourself from the Adamic tree. Having said so, cells must be brighter than that so that they can pass their genetic material to the daughter cells without necessarily compromising on amount. The logic behind that is that any cell that needs to divide must therefore replicate its DNA. If you remember the concept of the cell cycle, any cell that goes beyond the G1 phase enters the S phase, during which it replicates DNA. After it has replicated its DNA, it means that it has 2 copies of the same genetic material. That genetic material usually then condenses to form visible chromosomes. And so when we see a chromosome at that point, when that cell enters prophase, each chromosome will be having 2 copies of the genetic material. One of the copies is therefore called the chromatid. And so the chromatid and the chromosome are higher levels of DNA organization, which we see when a cell is dividing. You don't particularly see it outside cell division, but when a cell is dividing, then we see this. Great, so this shows you a chromosome with 2 chromatids. So this chromatid is the sister chromatid. This is a copy of the other one. Right, so those are the levels of DNA organization. That's just a recap so that you can link today's class with last week's class. Let's say some things about the normal human chromosomes. The chromosomes are numbered from number 1 to number 23. Well, usually we don't number the 23rd one. We usually just number the first 22. Then the 23rd one, we don't number them per se, but we call them sex chromosomes. So that the name given to the first 22 is according to the numbers, number 1 up to 22. But the sex chromosomes, we don't give them the number, we just call them sex chromosomes. But that means that we have 23 pairs of chromosomes. If a cell is diploid, it will have the 23 pairs of chromosomes. This represents 2 sets of chromosomes, one set having been obtained from the mother and another set having been obtained from the father. And so a diploid cell has 46 chromosomes in total. A haploid cell, on the other hand, will only have 1 set of chromosomes, not in pairs, just 1 set of chromosomes. And so haploid cells can only be seen in the germ cells, the sperm and the oocyte. The term homologous chromosome is therefore worth noting. Homologous chromosomes are chromosomes which carry similar genetic material, except that they're of different parental origin. So for example, here, these 2 chromosomes are homologous chromosomes. One was paternal origin, the other perhaps maternal origin. So usually these homologous chromosomes usually pair during metaphase 1 of cell division during meiosis. And it is because of that concept that now chromosomes can actually be karyotyped. And so I want to get it clear that karyotyping occurs, is done on cells which are at metaphase 1, because at that point that now they're in equator, they align themselves in the equator of a cell, and they exist in pairs in that particular stage. You cannot karyotype them at any other time because at that other time they are not aligned. The first 22 pairs of chromosomes are the ones we call autosomes, while the 23rd pair, the ones we call sex chromosomes because they code for gender characteristics. The sex chromosomes can either be X or Y chromosome. Great, that brings me to the first question that I want to ask you. So this question, I want you to just write on the chart there. I have put the chart in such a way that I'm the only one who will be able to see what you've written. So what I want you to write on this chart, I want you to look at these two karyotypes. The one on the left, let's call it A, and the one on the right, let's call it B. Just indicate whether they are normal or abnormal. So you can say both are normal, both are abnormal, or you specify which one is normal, either A or B, which one is abnormal, either A or B. So do that in the chat. Everyone, you have 30 seconds for that. There are a few people who have, however, gotten them wrong. So remember, these are both normal. The only difference is that this one is 46XX, so genetically female. And the other one is 46XY, so genetically male. Others are both normal. That brings me to another question. This one you'll have to think before you answer, but you still have 30 seconds. That's the question. So in question 2, you'll just choose the correct response there. Just write either A, B, C, or D. Again, you have 30 seconds. This one, we've gotten a lot of variation in responses. There are those who have gone for male phenotype. There are those who have chosen female phenotype. A few have gone for both male and female. So let's start with the, at least I'm happy nobody chose D. Let's start with anyone who chose C. Tell us, why did you think about that? So here we have Teresia. You're among those who chose C. Teresia, tell us, why do you think, why did you choose C?
Okay, just a trial. I was just thinking, just like the way like the normal karyotype up to 22. Now there is that aspect where we've got like maybe 2 chromosomes that has got XX, and also could be another one like 1 chromosome over Y. Like I don't know how to do it, but just the way it is usually displayed.
Okay, I hear you, no problem. Let me pick one person who chose B. Eunice, you're among those who chose B. Can you tell us why you chose B?
Okay, the reason I thought it could be a female, I thought about the XX as a pair. So to me, it appeared like it was overtaking the XY. So that's why I chose a female as an XX.
Okay, fair enough. That's good. There were many people with B, so let me pick 2 people. Dorcas, you also chose B. Why did you choose B?
Probably I thought that this would be a female with some expressions of a man, characteristics of a male.
Okay, I get you. Now that those who chose A, let me pick a few people. Mercy, you're among those who chose A. Can you tell us why you chose A?
I chose A because I remember the previous class, you told us that it's the absence of the Y chromosome that makes one female. So as long as there's a Y chromosome, that person is phenotypically a male.
Okay, fair enough. For you to get the correct response to this question, I'll have to show you the next slide. This slide tells us how sex is determined before birth. Prenatal sex determination. You may not know this, but there are 3 levels of sex determination before birth. And so there are 3 types of sexes that the fetus display or the embryo display. And they are established in sequence. One type of sex influences the next step of determination. So it is very strict, it is time dependent, very stringent, and one influences another, yet they are very different. The first type of sex is what we call the genotypic sex. The genotypic sex is also called the chromosomal sex. This, we just ask ourselves, which kind of sex chromosomes are present in the cells of this particular zygote or embryonic cells? So there could be 46XX, that is considered genetically female. And it could be 46XY, and that is considered genetically male. The big question is, at what point is the genotypic sex really established? The genotypic sex is determined at fertilization. It depends on the type of sperm that fertilizes the oocyte. So if the oocyte is fertilized by an X sperm, the zygote will be 46XX, genetically female. If the oocyte is fertilized by a Y sperm, the zygote will be 46XY, genetically male. Genotypic sex, which is also called chromosomal sex, is therefore determined at fertilization. Then comes the gonadosex. Before I tell you the gonadosex, it's important that you understand that there's something as the embryo grows. You know, initially it's just a zygote, then morula, then blastocyst, bilaminar disc, trilaminar disc, what we call gastrula, and things go on. At that time, there's no anatomical differentiation to a particular gender. At that point, the first few weeks of development, the embryo usually has a common structure that has the bipotential capacity of either becoming a testis or an ovary. That bipotential structure is called the indifferent gonad. any embryo will have the indifferent gonad. The indifferent gonad can either become the testis, therefore we call that gonadally male, or it can become the ovary, then we call that gonadally female. Question is, what makes the indifferent gonad form the testis and not the ovary, or form the ovary and not the testis? The determination of gonadal sex occurs in the period between 6 to 10 weeks of gestation, and it can only happen during that season. What makes it become the testis or the ovary? It relies on the presence of the SRY gene. SRY gene stands for sex determining region of the Y chromosome. And that is why last time I told you it depends on the presence of the Y chromosome. Because the presence of the Y chromosome would mean the presence of the SRY gene. When the SRY gene is present, it codes for a particular protein, which we call TDF. TDF stands for testicular determining factor. So this testicular determining factor is the one that influences the indifferent gonad of the embryo to become the testis. Therefore, in a situation where the baby is 46XY genetically male, the SRY gene will be present, and so it will code for TDF. And so TDF will direct the indifferent gonad of that embryo to become the testis. Things are aligned. If that is not the case, like in the situation of 46XX, there is no TDF, so there's no SRY gene because there's no Y chromosome. And so there's no TDF. In the absence of TDF, the indifferent gonad becomes the ovary as a default pathway. So formation of the ovary is a default for the indifferent gonad unless there is TDF. That is how gonadal sex is determined. Then the third type of sex is called the phenotypic sex. The phenotypic sex tries to answer the question, which kind of reproductive organs other than the gonads are present? Which type of genitalia do we have? Is it the male genitalia, where internally you have the prostate, seminal vesicles, vas deferens, and the like, and externally there is the scrotum and the penis? Or is it the female genitalia, where externally you have the vulva, and internally you have the uterus, fallopian tube, vaginal canal, and the like? On that note, I also wish to tell you that there's a time of development when the genitalia, either external or internal, is not committal to any particular anatomical entity with regard to gender differentiation. At that time, we call that the indifferent genitalia. So the indifferent genitalia can either become the male reproductive organs or the female reproductive organs. The determination of the phenotypic sex, which is also called the anatomical sex. The anatomical sex is the phenotypic sex. The determination of the phenotypic sex occurs a little later. It lags behind with a few days from the gonadal sex. I gave you a range there, so this is a range therefore. It depends on the presence of significant androgen hormone activity. Significant androgen hormone activity. You must have adequate amount of androgens for something to happen. If the amount of androgens are not adequate, something else takes place. Now, significant androgen hormone activity causes virilization of the genitalia. Therefore, the genitalia become male. In the absence of androgen, by default, the genitalia becomes female. Now remember, if the androgens are not adequate, by default, the estrogens will be high because remember it's during pregnancy. So estrogens are always there. So in the absence of significant androgen levels, that will be coupled with high levels of estrogen that will lead to development of female genitalia. But if you have enough levels of androgens, that will give us virilization leading to male genitalia. So the big question is, therefore, what makes this embryo have enough amount of androgens? For you to answer that question, it is important that you understand where sex hormones come from prenatally. There's some 4 structures that can produce hormones. One is a maternal ovary. But remember, the predominant hormone from the maternal ovary is estrogen, so it's not an androgen. The second structure is the placenta. Again, remember the placenta produces estrogens, so again, not androgens. The third structure is the adrenal cortex of the fetus. It does not matter the genotypic and the gonadal sex of that particular fetus. It doesn't matter. The adrenal cortex of the fetus produces androgens. It doesn't matter whether they are genetically female or male or gonadally female or male. The adrenal cortex of the fetus produces androgens, male sex hormones. So that's an important source of androgens. The other important source of androgens is the fetal gonad. The fetal testis produces androgens as well. The fetal ovary does not produce any hormone. It is hormonally silent. So don't be tempted to say it produces estrogen. It doesn't. But the fetal testis produces androgens. So if this pregnancy is characterized by production of androgens from the adrenal cortex as well as the fetal testis, the levels of androgens are going to be significant to cause virilization. However, if this pregnancy is not characterized by the presence of the testis, then the androgens that come from the fetal adrenal cortex are not adequate enough to cause virilization. And so it therefore goes, if the embryo is 46XY, genetically male, that embryo will eventually develop a testis, gonadally male. And that embryo will therefore have significant androgens to cause virilization, anatomically male. That is synchronous. If then you have 46XX genetically female, there is no Y, therefore gonadally this will be ovary. And that means that the androgen production will only be relying on the adrenal cortex. That will not be adequate for virilization. Therefore, Usually, anatomically, if there is no virilization, the genitalia will be of female phenotype. So with that explanation, you now understand why in the previous question, if somebody has Klinefelter syndrome, XXY, they'll be anatomically male. They'll not be anatomically female. They are not hermaphrodites. They are anatomically male. With that explanation I've given you. Understand, therefore, that you can have various disorders of sexual development based on this concept I've given you. They can be caused by either defects in the sex chromosomes. So maybe there's a Y chromosome, but the SRY gene is defective. It has mutated. That could give problems. So defects in the sex chromosomes. It could also be caused by excess androgens in an XX embryo. If you have excess androgens in an XX embryo, for example, if the fetal adrenal cortex of this female embryo is hypertrophic, so that's a context of pathology, it's producing a lot of androgens. The levels of androgens can be significant enough to cause virilization. Or if this female is lacking the enzyme that converts androgens to estrogens, so they cannot synthesize estrogens, but they are synthesizing androgens, the levels of androgens will accumulate a lot to cause virilization in those babies. Similarly, if a baby is 46XY, so genetically male and perhaps gonadally male, but there's a problem. The androgens are not working. That can cause this baby to become female anatomically. Even though genetically they are male, they'll be anatomically female. Those are disorders of sexual development. We'll revisit them perhaps one of the last lectures we'll have this particular semester. So you understand why the previous question, the answer will be male. But then I want to give you another question. So gonadodysgenesis, if gonadodysgenesis occurs in an embryo who is 46XY, what do you think will be the anatomical sex of that particular baby? Okay, you have another 30 seconds. I'll call that question 3. So you have 30 seconds to answer, you carry on. This one seems to have given you people trouble, but Maureen, you said B. Tell us why you said B.
Okay, I said B because you say it's dysgenesis. When you talk about dysgenesis, like I think the androgen is less, and this causes when there's an absence of androgen, that makes the person anatomically female.
Good, fair enough. So dysgenesis here means that the gonad has not developed. So this baby does not have a testis, does not have an ovary. Yes, in that context, Maureen, you're right. We can't have significant androgen hormone activity in such a baby. So genetically, they're male, but anatomically, they'll be female, even from birth, and you'll not know. You might just see some things now at puberty, but at birth, you won't know. You'll even give them a female name. Okay. Another question. If two sperms fertilize a single oocyte, we call that polyspermy. What would be the outcome of polyspermy? I'll call that question 4. Again, 30 seconds for you. I've closed it now. Let me hear from those who have not responded. So I'm trying to pick who has not responded. Esther, you are among those who have not responded. Esther, can you hear?
Yes, I can.
Okay, you didn't respond, but tell us what you think.
I think it's A, but I was so confused
Why do you think it's A?
because the two terms were like, I don't know.
The two sperms?
The oocyte will divide or something. I really don't know why I think it's A.
Okay. I'll give it to Bole. Bole, you also didn't respond. Bole, Ado? Carry on.
Okay, thank you. I was trying to think through because I'm not sure though about two sperm fertilizing single oocyte. I know it is one sperm for single oocyte, so I'm not quite sure. So I was trying to think through.
Okay, so you, in your opinion, the question was a wrong one, eh? Okay. One last person, Teresia, you also didn't answer. Terry?
Yes. I was also trying to think through, just as the way Bola has mentioned, because I'm just finding if there's going to be some maybe faulty kind of segregation of the chromosomes. Yeah, so I was still thinking about it.
Okay. Now, interesting enough, only one person got it right, and that person is Suada. Suada, you got it right. Tell us, what was your thought process?
My thought process was that twin pregnancy is whereby one ovum is being divided first, then triplet is where one ovum. When I see all these answers, I saw it is none of the above, because when it is abnormal sperm, it is an abnormal genetic disorder that will show up. It will not be monosomy, and it will not be trisomy, and it will not be triplet and twin. Then I took none of the above.
So why is it not trisomy? Most people wrote trisomy. Why is it not trisomy? Tell them why they are wrong.
Trisomy, they have an extra chromosomes at 21. That is a Down syndrome. And Down syndrome is one ovum and one sperm being fertilized.
Okay, the trisomy syndrome doesn't have to be trisomy 21 though. It can be any trisomy. Anyway, let me look at it this way. If a trisomy syndrome has 47 chromosomes in total, that's a trisomy syndrome. But if you have two sperms fertilizing a single oocyte, how many chromosomes will the zygote have? Mtega, how many chromosomes will the zygote have?
So it will be 23 times 3.
And that would be 69.
69, yes.
So 69 is not a twin, neither is it a triplet, neither is it a trisomy. It's a genetic disorder whose answer is not on this list. And so none of the above is the option. But anyone who knows, if somebody has 69 chromosomes, what will that become? Any volunteer? What will this genetic disorder lead to? It's something you all know, by the way. Okay, let me take it up. It will lead to molar pregnancy. That pregnancy will become molar pregnancy, hydatidiform mole. Now, you see, molar pregnancy is a pregnancy characterized by vesicular proliferation of placental tissues. So the placenta, instead of forming the normal placenta, forms vesicles. These vesicles may have some fetal parts or may not have fetal parts. The cause of molar pregnancy is if you have an extra set, or let me say excess set of paternal genome. We are comparing with the maternal genome. Now, understand the context here. The normal setup is that you have 23 chromosomes, that's one set from your father, and one set of 23 from your mother. So both sets, just one, so there's equal. Maternal one set, paternal one set, so it's equal contribution. We get molar pregnancy if the contribution from the paternal genome is more in terms of the set of 23. So if you have extra set of paternal genome, you'll get molar pregnancy. There are different mechanisms for that, and I can take you through it quickly. One is polyspermy. If a single oocyte is fertilized by more than one sperm, let's say that a single oocyte is fertilized by 2 sperms, that will lead to what we call a triploid state. Now, not a haploid or a diploid, but triploid. So triploidy would have been one of the answers if it was in those choices. A triploid state, 69 chromosomes, but not trisomy. So distinguish trisomy and triploidy. Well, the karyotype of the zygote will be any of those 3 options, depending on which type of sperm fertilizes the oocyte. If the oocyte is fertilized by 2 sperms which are both X, then that's what you have. If the 2 sperms are both Y, this is what you'll have. If the 2 sperms are different, then this is what you'll have. This type of molar pregnancy will give us what we call partial molar pregnancy. In partial molar pregnancy, there are some fetal parts present. The second mechanism is that an oocyte may be fertilized by 1 sperm, but the sperm that is fertilizing the oocyte replicates its DNA. It enters into the S phase before the fusion of the genome occurs. If that happens, the zygote will again have a triploid state, except in this case, the karyotype can only be 69 triple X if the sperm was an X chromosome, an X sperm, or 69 XYY if that single sperm was a Y sperm. You cannot have 69XXY in this case. Either way, this one will still give you partial molar pregnancy, where we have vesicular proliferation of placenta, but we also have fetal parts present. The third mechanism, the third cause of molar pregnancy is a unique one. This one, an oocyte may be fertilized by either 1 or 2 sperms, but there's a condition. The oocyte being fertilized is genetically empty. What that means is that there's no genetic material in that cell. The cell is anucleate. It doesn't have a nucleus. Maybe the nucleus got destroyed or something, but it is not genetically, it doesn't have the maternal genome. So if that happens, if it is fertilized by 2 sperms, nothing much will happen to the sperms. But if it's fertilized with only 1 sperm, the single sperm usually replicates its genome. Then the resultant cell, therefore, will have a total of 46 chromosomes. It might appear normal, but the abnormal thing about it that everything is of paternal origin. There's no maternal genome in this particular zygote. This type of pregnancy is the one that gives complete molar pregnancy. In complete molar pregnancy, there's no fetal parts. So it's just placental tissue only, no embryonic parts. Okay, so I hope you've understood the concept of triploidy well. Don't confuse it with the trisomy. I don't know whether there's any question related to that background before I go to greater detail about the DNA now. So let me give you a chance to ask if you do have. This second agenda.
Somebody who has asked that maybe I think she has not understood or the concept that you said earlier. It's in the chat box.
Oh, yeah, I saw it, but I didn't answer it because the person didn't log in with the official names as we agreed. So I don't know who that is.
Okay.
That's why I didn't answer it.
I think it's Monica.
Okay.
Because she had some problem in logging in.
Okay.
And at the end of the day, she succeeded logging by that name.
Okay, no problem. So Monica, what you're saying is that there are 2 types of sperms that will determine the type, the gender of the baby, the Y sperm and the X sperm. The Y sperm swims faster, but doesn't live long within the female tract. The X sperm swims slowly, but lives longer within the female tract. So if you want to conceive a female child, you need to increase the probability of it having only X sperms at the time of fertilization. So you have sex some 3 days prior to ovulation. But if you have sex on the day of ovulation, you'll be giving the Y sperm an upper hand because the one that swims faster. If you have sex 3 days to ovulation, you'll be giving the X sperm an upper hand because the one that lives longer. That's a concept. Okay, I hope that's clearer now. We can now go to the next agenda. So the next agenda is to talk about the basic structure of the DNA. The DNA exists as a double-stranded molecule. So these are 2 strands which run side by side. We call these strands complementary strands. A single strand is made up of a series of what we call nucleotides. one single strand is made up of a series of nucleotides. What does a nucleotide consist of? A nucleotide consists of 3 molecules. There's a phosphate group. There's a deoxyribose sugar. And there is a nitrogenous base. So those 3 make up a nucleotide of the DNA. Now let's look at that. Consider these 3 to be the 3 components of a nucleotide. That yellow one represents the phosphate group. I don't know whether to call that red, but it represents the deoxyribose sugar. And then this other one represents the nitrogenous base. Those are the 3 components of a DNA nucleotide. Now we are saying a single strand consists of a series of nucleotides, which means you have another nucleotide, another one, and yet another one. So one strand will be just a series of nucleotides, interconnected. Important to note is that the backbone of this series of nucleotides is made up by alternating phosphate and deoxyribose sugar. Alternating. So phosphate, deoxyribose, phosphate, deoxyribose. That's the backbone of the molecule. The nitrogenous bases, on the other hand, form the teeth which the strand is going to use to pair with the other complementary strand. So the other complementary strand will also be looking like this, with these bases facing one another so that the bases can pair. The nitrogenous bases are the ones which are used for pairing. So this image shows you that concept. If this is one strand, this is the other strand. So we have alternating sugar and phosphate group on the backbone, and then the nitrogenous bases for the teeth. You can see these bases for this molecule and these bases for this molecule. They are pairing there. So the bases pair. Phosphate and sugar form the backbone. Now, I've already told you that statement that the 2 DNA strands pair using the bases. Let's talk about how the bases pair. There are 4 bases. We have what you call adenine. It pairs with thymine. If this was RNA, instead of thymine, we have uracil. Guanine pairs with cytosine. So those are the 4 nitrogenous bases which are found in the DNA. And that is how they pair. You can use the letters to represent what they are. So guanine with cytosine, thymine with adenine. So that's the basic structure of the DNA. Remember what I told you, the levels of organization. So the DNA will be those double strand forming the helix, then the nucleosome, chromatin, things like that. If this cell needs to divide, it has to replicate. So let's talk about DNA replication. DNA replication, basically the duplication of the DNA material. This occurs during the S phase of the cell cycle in cells which are inevitably going to divide. So the first thing that must happen to the DNA is that the DNA must first unwind itself. That's the unzipping, that it must unwind itself. The 2 strands unwind. So it undoes the helix structure. After it has unwound, each of the strands become a template for the formation of a complementary strand. So the older strands form the template for formation of newer strands. Each of the old templates make new strands. So the third image there shows you how the new strands are being formed from each of the old templates. Now understand, therefore, that if we consider strictly the concept of base pairing, then this strand here, this new strand that's being formed, will actually be very similar to this one. And this strand that's being formed will be very similar to that one. So in a nutshell, the two molecules will actually be similar. In DNA replication, the two molecules must eventually be similar. The process of DNA replication is guided by an enzyme, DNA polymerase, that usually guide the process. The new molecules which are formed are actually identical, they are similar. That is how DNA replication takes place. Remember, it has to be in a cell that's about to divide. Now we can define a gene. What is a gene? Commonly defined as the molecular unit of inheritance. A gene refers to the DNA base sequence that codes for the type and order of amino acids of a single protein. The base sequence, which means that if you are to remember that DNA sequence, okay, let me show you this one. So if in this particular DNA molecule, this is one strand, this is another strand, let's take one of the strands. So here we see G, G, C, G, A, A. That is the base sequence I'm talking about, because these are the bases, guanine, cytosine, and the like. So the sequence of bases here that code for the order in which amino acids appear in a particular protein is what we're calling a gene. The base sequence that code for the type and order of amino acids in a single protein. Remember that proteins are made up of amino acids. Usually, a sequence of 3 bases represents one amino acid. So perhaps this GGC is one amino acid, GAA is another amino acid, CGC is another amino acid, and the like. And you just read them in that order. If this was RNA, that sequence of 3 is called a codon. That sequence of 3 in an RNA is what we call the codon. It still represents a single amino acid. Having said so, let's remind ourselves, therefore, about the process of protein synthesis. Then we stellate with that particular thing we mentioned right now. So last time we talked, we said that protein synthesis occurs in some steps. One, there must be a process of transcription, where the messenger RNA is formed from the DNA template. So what usually happens is that if you need a single amino acid, you'll just form an RNA for that. If you need a single protein, you'll form an RNA for that particular protein. The DNA molecule contains many genes, which means contain many proteins. But an RNA can only carry information of one single protein. So let's say this cell needs insulin. The gene for insulin is located in one of the chromosomes. That particular gene will open up first, then a messenger RNA copies the gene for insulin. So you form RNA. That's the process of transcription, the process of formation messenger RNA from a gene sequence. That will take place within the nucleus. It's guided by what we call the RNA polymerase enzyme. After you form the RNA may undergo some processing though, and then it enters the cytoplasm. When it enters the cytoplasm, it will go to the ribosomes, where now it will be responsible for giving the information for formation of proteins. And that's the one we're calling translation, the process of formation of polypeptide chain from the RNA code. So remember, there is a messenger RNA code that has coded the order or sequence of amino acids as they should appear in a particular protein. That code will be read 3 bases at a time, which means each codon at a time. And then amino acids are formed based on that codon. It's about changing the genetic language to phenotypic language. That's what we're calling translation. After translation, there are other processes. Remember, we have post-translation modification, we have packaging, and exocytosis. Now, let me say something more about the reading of the codon now. This is not for your cramming, it's just for your illustration. Let's take, for example, that this is an RNA molecule that should be used to form a protein. So the order in which these bases appear will inform the order in which amino acids will be formed in a particular protein. If you take, for example, GCU as one codon, we can read here GCU, that is alanine. So that's the first amino acid there. Then the second amino acid is ACG. We can go to ACG, we see threonine there as the second amino acid. The third amino acid is GAG. So let's look at GAG, so that's glutamate there. Then CUU, that's leucine. Remember, U is for thymine, so in RNA we have uracil instead of thymine. So that's leucine there. So you see the order of amino acids that are being formed on this particular protein are informed by the order of the bases that are present in the RNA, which were informed by the order of the bases that appear in the DNA. And that is why the DNA is the one that stores the genetic material. The information is stored in the DNA, but now just being translated here. So as I said, it's not for your cramming. Now you go cram what is AAG and things like that. Not necessary, even I don't know these ones. So I have to refer to the code for me to get an interpretation. Right, so after translation, we have post-translation modification, which occurs in the RER and the Golgi apparatus. Last time we said that this post-translation modification involves changes that occur to the polypeptide chain. After post-translation modification, we have packaging, which takes place within the Golgi apparatus as well. The proteins are packaged in vesicles. After packaging, if the protein needs to be released outside, then it will have to undergo exocytosis. So this will be from the secretory vesicles, the walls merging with the cell membrane, and then the contents of the vesicles released into the extracellular space. That's the process of protein synthesis. Now I brought that so that we can then understand what is gene mutation and the mechanisms and outcome of gene mutation. That's why I chose to remind you protein synthesis and see then the importance of the gene. That the importance of the gene here is to code for proteins which are being formed. in an individual. It codes for proteins. Remember, these proteins could be structural proteins like melanin, hair, but could also be enzymes, for example, or hormones. So it codes for proteins. Back to now gene mutations. What are gene mutations? Gene mutations are alterations in the normal base sequence of a gene. You may have a normal sequence of a gene, then some alterations occur to it. Those alterations are the ones you're calling mutations. Mutations can either be what we call point mutations or what we call frame shift mutations. What are point mutations? Point mutations are alterations which occurs in the gene sequence involving one base pair without changing the reading frame of the gene. If you remember what I showed you, that other previous example, the reading frame, or we can use this one. Let's take this to be the reading frame. I know you read as cut, but let's say CAT, 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 CAT. So CAT represents a particular amino acid, maybe threonine. This particular amino acid, so it's just repeated amino acids. We are using CAT for the sake of understanding easily, but understand that 3 of them represent one amino acid, CAT, CAT, CAT. So that's a normal protein. The order of amino acids will be normal for this particular one. In point mutations, we are changing only one base pair without necessarily changing the reading frame. For example, here, we had CAT, 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 but you see at position cytosine there, it has been changed with the guanine. So you may have that kind of change at a single point. It doesn't change the rest, the others remain the same. So those are point mutations. We call it inversion if the base pair has just altered. For example, we say that that guanine usually pairs with cytosine. So in this case, cytosine has been changed to guanine. This is an inversion. We've altered what was in the complementary strand has come this side, and what was this side has gone to the complementary strand. That is inversion. Substitution is if you change the base pair completely. So instead of perhaps having G or C, you have A or T. That's a substitution. Either substitution or inversion give you point mutations. Then there are those that we call frame shift mutations. Frame shift mutations are caused by either deletion or addition of a base pair. Remember, this is the original strand. If we add something here, addition, or we delete something, we'll change the reading frame. Why do we say so? Look. So ours, in this case, is a deletion. So the original one was CAT, then was supposed to be CAT, but T has been deleted. So the next amino acid will have to be CAC, and then ATC, ATC, ATC. You've changed the reading frame completely just because of that deletion. That will also happen if there was an addition. If you add something perhaps here, you will change the reading frame completely. So deletion and addition mutations, mechanisms lead to frame shift mutations. frameshift mutations change the product completely. It changes the product completely. So that if it was, let's say, hair, it can change to something totally different. Unlike point mutations where the protein will still remain, except that might just be a defective protein. It could be melanin, but a weaker form of melanin. So frameshift mutations are worse compared to point mutations. But from a medical point of view, you'll be more worried by point mutations than frameshift mutations. I know you're wondering why. Now, the reason is because frameshift mutations are so bad that it doesn't lead to anything. The cell will die most of the time. It is so bad that that cell will be caused to die. Or it might not lead to formation of anything at all. It's lethal. But point mutations may not be lethal. Because they're not lethal, now these ones will have some clinical symptoms in an individual. And that's also the concept of cancer. We'll see that next week. That the mutations that occur that cause cancer are mutations which are not lethal. That's why they then lead to abnormalities. Because if they're lethal, they lead to the death of the cell. And so there'll be no cancer. Okay, having said so, let's look at this. Still the same example I gave you. So CAT, 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 CAT. If you look at this CAT, so CAT, so T means U, that means histidine. So this is a protein that is just formed of histidine throughout. Let's say that's the normal one. If you have a point mutation, so we'll have CAT histidine, but then the second amino acid there will not be histidine. Instead, it will be, I'm reading that, it will be aspartic acid. So histidine, aspartic acid, then histidine throughout. It will be the same protein except just an alteration in one of them, and so slightly functionally defective, but it's still there. Compare with this one. If you have a deletion there, so we'll have a CAT histidine. The second amino acid is CAC. Okay, so coincidentally, this is still the same histidine, no problem. But what's the third amino acid? ATC. So ATC means leucine. So the rest of the frame will just have leucine throughout. Now that's why we're saying frameshift mutations have more detrimental outcome than point mutations. So what are the causes of gene mutations? Gene mutations can be caused by errors in DNA replication. When the DNA was replicating, there were some errors that did not. Okay, let me just put that way, that there can be errors during replication. There's some chemical agents that can also cause gene mutations. We can have radiation, X-rays, or gamma rays can cause mutations. And lastly, viruses can also cause mutations. I can see Sadia with a question. Sadia, carry on. Sadia.
On the previous, yeah, on the previous slide, what will happen maybe physically for people who have the abnormal when there is mutation, maybe when there is abnormal arrangement of the proteins, of the amino acid, I mean.
Okay, so it could form a totally different protein in the body. Totally different protein in the body. So it will depend on the kind of mutation that has occurred. Or most of the time, you know, some of these codes, if you check carefully, some of this code means stop reading. So if accidentally then the mutation leads to a code that reads stop, it means that that amino acid is not produced at all. So it becomes a shorter chain of amino acid. So it will depend on the mutation itself. And there are millions and millions of proteins. So we can't specify what exactly will happen in which particular mutation, but it will depend on the mutation itself.
Okay, thank you.
All right, Karibu. So the causes of mutations, I've already told you this before, but let me also give you the outcome of mutations. What's the outcome? If a mutation takes place, most of the time, the DNA will be able to repair itself. Most of the time, the DNA repairs itself. However, if the DNA does not repair itself, the cell might be forced to die in that sense. And those are check mechanisms available. And so if the cell dies, that again is okay. Remember, just a single cell, it's not the whole person dying, it's just a single cell. However, if the cell does not die and the mistake is not repaired, then that mutation is made permanent. When that cell divides into daughter cells, it becomes permanent. It becomes the new normal for that particular cell. So mutations are usually made permanent by DNA replication. If that cell does not divide, it will not have to replicate. So there's nothing, it's the end of that particular cell. But if there's DNA replication, it means that that cell is going to divide. And so depending on the number of divisions that that cell will have eventually, you'll have mutations taking down to even future generations. These mutations can be carried on to offsprings if the mutations involve the germ cells, the sperm and the oocyte. So if they just involve the somatic cells, you cannot pass on that to your babies. But if the mutations involve the cells, the gamete cells, then you can possibly pass that mutation to your offspring. And that explains the concept of familial inheritance of some genetic disorders. There were disorders that happened on the germ cells and then eventually transmitted to the offsprings. And so you might find that the parent doesn't really have the disease, but now the children have the disease. Okay, now let's classify genetic disorders and give some common examples. We can classify genetic disorders depending on the cause. Some of them are caused by chromosomal defect, the defects in the chromosome. And some of them are caused by defects in gene mutations. The chromosomal defects are classified into two. You can have numerical chromosomal disorders, which we call aneuploidy syndromes. And we talked about some of them. We talked about them last week. It can be trisomies when you have 47 chromosomes, or monosomies when you have 45 chromosomes. Those are numerical chromosomal disorders. Then you can have structural chromosomal disorders. The number of chromosomes is okay, but the structure of the chromosome has a problem. Some disorders related to this include Fragile X syndrome. You can check on that. And Crudisha, which means cry like a cat. It's also another disorder. You can check on that. They are more functional than anatomical. Then the disorders caused by gene mutations are classified based on the chromosome that houses that particular gene. Gene mutations can either be autosomal disorder, if the gene is housed by the first 22 chromosomes, or sex-linked disorder if the gene is housed by the 23rd pair of chromosomes. We can also classify gene mutations as being dominant or recessive. Dominant means that You just need a single inheritance, either from the mother or the father, for it to show. And recessive means that you must inherit it from both of your parents for it to show. So you have dominant disorders and recessive disorders. Now, in terms of how we classify them, we combine the first concept, the second concept to give us some terminologies. For example, you can have autosomal dominant disorders. You can have autosomal recessive disorders. We can have X-linked recessive disorders, just to give examples, but you can have combinations. Perhaps X-linked dominant, Y-linked dominant, Y-linked recessive as well. But these are the ones you'll come across often, and these are the ones I'll give you examples about. Let's start with the numerical chromosomal disorders. We mentioned it last week, though. Numerical chromosomal disorders are caused by non-disjunction of chromosomes during meiotic cell division, which means the division that occurs within the gametes. So if you have non-disjunction of chromosomes, it means that the daughter cells will have either an extra chromosome or lacking one chromosome. Remember, the gametes need to be haploid. So it can be a haploid cell with 24 chromosomes or a haploid cell with 22 chromosomes if there is non-disjunction. If a haploid gamete with 22 chromosomes participates in fertilization with a normal gamete, that will give you a monosomy syndrome. And remember, we mentioned that the only monosomy syndrome compatible with life is Turner syndrome. If a gamete with an extra chromosome participates in fertilization, that will give you a trisomy syndrome. A trisomy syndrome will have 47 chromosomes in total. Even though I gave you a whole list of aneuploidy syndromes, it is important to note that most of the aneuploidy syndromes are not compatible with life, and so they terminate as miscarriages. As a matter of fact, close to 50% of conceptions usually lead to terminations, early pregnancy loss. Some of them, you may not even know that you conceived. Actually, most of them, you may not even know that you conceived, because they'll just last the first few days after conception. Even before you miss your period, the conceptus has already involuted because of genetic disorder. So majority of aneuploidy syndromes terminate as miscarriages. Now, you need to understand the mechanisms of aneuploidy. It is due to non-disjunction, yes, but in which phase? So this is another question for you. You will answer that in the chat. This is question 5. You have 30 seconds for that. Another question. So if you have a trisomy syndrome of 47XYY, at what point did the non-disjunction actually take place? Let me make your work simple. Is it anaphase 1 or anaphase 2? We've already agreed that the non-disjunction occurs at anaphase. So forget about A, B, E, and F. It is either anaphase 1 or anaphase 2. Okay, that makes your work easier, so you have 30 seconds again. I think these ones I'll be quick because we covered them last week. Aneuploidy syndromes can be of different types. So to your left, that is trisomy 21, which is Down syndrome. To your right, that is 47XXY, which is Klinefelter syndrome. Last week, we agreed on why you call this trisomy 21, but why we don't call this trisomy 23 Instead, we use letter 47 to mean trisomy, then give us the pattern of the 23rd pair. If you have 45X0, you call it Turner syndrome, the only monosomy syndrome compatible with life because all autosomal monosomies are lethal, and also complete absence of the X chromosome is not compatible with human life. Triple X syndrome is 47 triple X or super female. You can also have trisomy 18, which we call Edwards syndrome. Trisomy 13 is Patau syndrome. So the most common aneuploidy syndromes are Turner syndrome, sorry, not Turner syndrome. Trisomy 21, that is Down syndrome, the most common, followed by these two, trisomy 18 and trisomy 13. So those are the most common aneuploidy syndromes. However, Edward and Patau are not so much good compatible with longevity. So the children die in early age. Down syndrome can move for some time, but Edward and Patau, they die at an early age. Things like Klinefelter may live throughout their life, actually. Triple X syndrome, XYY syndrome, those ones will live long. To your left here is Jacob syndrome, which is XYY or super male, who can actually live up to adulthood. Trisomy 8 is a bit lethal as well, so maybe just a few months of life. Okay, so those are numerical chromosomal disorders. Now let's talk about the gene mutations, autosomal dominant disorders. When we say something is autosomal dominant, the autosomal here means that it affects the first 22 chromosomes. So the gene affected is located within the first 22 pairs. And then we call it dominant to mean that a single copy of the mutated gene is enough to give the disorder. You don't have to inherit it from both parents for it to give you the disorder. Just a single copy will give you the disorder. So usually if somebody has autosomal dominant disorder, the children have a 50% chance of being affected. The offsprings have a 50% chance of being affected. Examples of autosomal dominant disorders are many. We can have autosomal dominant polycystic kidney disease, Digeorge syndrome, which involves perhaps congenital absence of the thymus and the like, hypertrophic cardiomyopathy, where the heart just enlarges and there's no identified cause. Marfan syndromes are connective tissue disorders. Polydactyly is where there's an extra finger. Achondroplasia is where the cartilage doesn't really grow well, and so the person remains really short. Aniridia is the congenital absence of the iris. So these are examples of autosomal dominant disorders. I can see a hand raised. Carry on, Sadia.
I was just wondering whether sickle cell fall in this category, sickle cell anemia.
Okay, sickle cell is not autosomal dominant, it is autosomal recessive. I'll come to that shortly.
Okay, okay.
All right. So this is what I mean by aniridia. Look at that eye. The pupil is so big because the iris is not really there or largely absent. That's aniridia. Achondroplasia, where you have an adult who is very short. I believe you've seen that kind of anomaly. Now, autosomal recessive disorders. Autosomal, we've already defined that. Recessive here means that 2 copies of that gene are necessary for the disorder to show. So you'll have to inherit one from your mother and one from your father for that to show. Those are recessive disorders. If you have only one of them, you are a carrier. you don't really show the disease, but you're a carrier of that particular disorder. Examples now be, that's where you put albinism, sickle cell disease, cystic fibrosis, Tay-Sachs disease. These are some storage disorders. So this one shows you for comelia, it's also an autosomal recessive disorder where there is absence of the intermediates. So the hand is attached to the trunk. This one shows you albinism. I believe you've seen that. How about sex-linked recessive disorders? I want us to think through these ones. Yes, Cecilia?
A question on the autosomal dominant for the achondroplasia. Not that it's the short adult. Can it be diagnosed in the early stages of development?
Yes, it can be diagnosed in early stages of development, but how early? Usually in childhood periods, basically. Of course, not at the time of birth, difficult at the time of birth, but you realize that this child is not gaining height as the other children. So perhaps within 1 or 2 years, you can be diagnosed that one. But that time, we usually just diagnose them radiologically. Okay. Right. So sex-linked recessive disorders. It means that the gene causing the disorder is housed in the X chromosome. We've already defined what recessive is. It is more important, it's very important for you to understand that statement there. I wish I asked it before I showed it, but it has already shown that if somebody has an X-linked disorder, or rather, let me put it this way, X-linked recessive disorders are seen more in males than females. Maybe I ask why. Anyone? Why do we have X-linked disorders more in males than females? It's the X chromosome. Anyone?
Just a trial.
Yes, Adia.
Yeah. Is it because they only have one X chromosome?
True, because they only have one X chromosome. You know, females will have 2 X chromosomes, and so the other X chromosome is protective. For males, as long as it is there, it will show. So it will be like a dominant trait in males, and that is why more men will have it. Females will be largely carriers. If a female has the trait, they'll be a carrier unless they get it from both parents. Now, I don't know how to ask this. Okay, let me put it this way. If a man has an X-linked recessive disorder, can he pass it on to his boys? Yes or no?
No.
Maybe that. Okay, no, yes, no. Okay, Daniel, give us a reason why no.
I think because during the sex formation, we only have involvement of the X and the Y chromosome. So if we are going to have a boy, we'll have an X from the mother and a Y from the father. But then this sex-linked recessive gene is attached to the Y in the father, so it's not going to be crossed to the boy.
Okay, it's attached to the X, so it's not going to be crossed to the boy. You're right. So that is important to note, that affected fathers cannot pass X-linked traits to their sons because fathers give Y chromosome to their sons. So examples of sex-linked recessive disorders include hemophilia, muscle dystrophy, ichthyosis, agammaglobulinemia. So maybe let me tell you something. Ichthyosis is where the skin looks like that one of reptiles with it's very scaly. A gamma globulinemia is basically absence of gamma globulins. So this is an immune disorder. SCID stands for severe combined immunodeficiency disorder. So these are congenital immunodeficiency syndromes. Remember, there is acquired and congenital. AIDS is acquired, caused by HIV, but we also have congenital immunodeficiency syndromes. So this severe combined affect both humoral immunity as well as cellular immunity. These are X-linked recessive disorders. The images here show you that of ichthyosis, how the skin looks like, and this one is hemophilia, bleeding tendencies. Okay, 2 more agenda to go. I'll be a bit quick on this so that we finish in good time. We want to see the interaction between genes and the environment. Now, genes and the environment interact this way. There are many diseases. These diseases are largely multifactorial. Most diseases are multifactorial, to mean that for that disease to show, you must have interaction between the genes and the environment. I may have a particular genetic disorder. It may not show unless there's an environmental trigger. Also the other way around. I may have some environmental triggers, but if there's no genetic predisposition, a disease may not show. So many diseases are actually multifactorial, that there's a genetic predisposition, and so there's an environmental trigger to that genetic predisposition. So there's interaction between the gene and the environment. So those genetic predispositions increase the risk for the environmental exposures to cause diseases. Left alone, the genetic disorder alone may not give you that disease, and the environmental exposure alone may not give you that defect. But when they work together, they give you those diseases even at higher levels. So that's the message I want you to get from that particular slide. Examples of diseases where the genetic disorders and environments interact, the list is really endless. This is just a few. And if you look at that list, the things you've seen, the things that you are familiar with. So a number of those conditions, there is the interaction between the gene and the environment. Right. Having said so, I want to now talk about epigenetics as I finish. We'll define what are epigenetics, and we look at the mechanisms of epigenetics. Epigenetics refer to modifications that alter the expression of a gene without necessarily changing the sequence of that gene. So understand, it doesn't cause mutation. It doesn't alter the gene sequence, so there's no mutation. The gene is still okay. The DNA is still okay. But there's some alterations that happen to it so that the gene is not expressed. When we talk of gene expression, we simply mean protein synthesis. So the gene is not available, or it's more available for particular formation of a particular protein. It alters the expression. It can either suppress or overexpress itself. Those are epigenetics. There are alterations, modifications that make a gene express itself more or less. Most often, this will involve changes that affect the activity, as I've said. So there are several environmental factors that can actually lead to epigenetics. So what are the mechanisms of epigenetics? Three mechanisms have been explained to explain epigenetics. One of them is the concept of DNA methylation. The DNA molecule can be bound with a lot of methyl group, usually on the cytosine, so that you have what we call methyl cytosine. Methyl cytosine will still function as a normal cytosine. It will still have the same kind of genetic information like the normal cytosine. The only difference here is that a heavily methylated DNA may not be easily available for transcription. So it will move the DNA from being euchromatic type of chromatin to the heterochromatic type of chromatin. So yes, the gene is there, but it is not being expressed as a mechanism. It's not being expressed easily because of DNA methylation. So that's one of the mechanisms of epigenetics. There are modifications that occur on the DNA affecting its expression. The other mechanism is modification of the histone proteins. The histone proteins are the proteins that hold the DNA. Remember, the DNA is highly coiled so that it doesn't break easily. That's the reason for high coiling of the DNA, so that it doesn't break easily. So the histone proteins are usually modified, again, with the addition of methyl or acetyl group onto it. And such histone proteins anchor the DNA more strongly. So there's some modification that can occur to the histone protein that make the histone protein not release the DNA. So that the gene sequence is there, yes, but because of the modification on the histone, the DNA is not easily released, it's not easily unwound. And so that will affect its expression. Last but not least, there's some mechanisms of epigenetics that revolve around the RNA, now not the DNA, but the RNA. Those RNA strands which are usually formed from the DNA, some of them have some alterations. They can still be methylation still, but there are alterations that affect the RNA, so that sometimes transcription has already occurred, but something happens to that messenger RNA, its structure is altered, so the protein will not be synthesized from it. Those are alterations which affect the RNA. So these are the mechanisms of epigenetics, the three major causes of epigenetics. What's the evidence of epigenetics in medicine as we finish? Think about monozygotic twins. So monozygotic twins means that they're genetically similar. Then take these monozygotic twins, look at them, follow them as they grow up. Initially, they look really similar, perhaps because they've been born in the same hospitals, subjected to similar things. But as they grow up, they become different in terms of even character, even what they score in school, even their liking, their reaction to anger, things like that. The type of diseases that affect them will even be different as they grow older. Because as they grow older, there's a lot of epigenetic interactions that occur, which now influence how they express themselves phenotypically. Think about what we call genomic imprinting. Genomic imprinting is the silencing of a gene depending on the parent that contribute to that particular gene. What I mean is that there's some genes that can only express if it came from your father. It won't express if it came from your mother. And there's some genes that can only express if it came from your mother. It cannot express if it came from your father. Usually there's a way that some genes are silenced depending on the parent they came from. Even if you have the trait, it will not show they're silenced. That is what we call genomic imprinting. So it occurs. And this explains why some particular gender will express some particular things consistently within the family line, and you'll not see it in the other gender. It doesn't mean that they are only located in the X and Y chromosomes, not necessarily. They can even be located in the autosomes, but some genes are silenced. You can also have some epigenetic alterations that affect DNA repair or that affect the cell cycle genes that control the cell cycle. If you have alterations on those genes, it can be bad because now this can actually cause alterations that even lead to development of cancers. We'll talk more about cancers next week. Think about addiction and drug use. People who use opioids for a long period of time, from a molecular level, it usually leads to expression of genes that are associated with addiction. And that explains addiction in those kind of situations. Now think about a diabetic who has a wound. You may not have never thought about this before, but there's association between hyperglycemic condition and the functioning of the macrophages. Macrophages tend to be more of inflammatory type when the environment is hyperglycemic. And so that explains also why we'll see chronicity of diabetic wounds, because the macrophages there are not releasing chemicals that are pro-healing for regeneration of cells. They're releasing chemicals which are for pro-inflammation. And that makes the wound even to continue for a long time. So can we say that those macrophages genetically are defective? No, it's because of the hyperglycemic condition that they've been put to. Great, so these are some of the examples. Just don't intend that you remember them, just put them here so that you see some evidence that alterations, modifications can occur on the DNA that affect how that DNA is expressed, and that's what we call epigenetics. Okay, thank you very much. Now I'm done, I'll stop there, but I'll give you a chance to ask questions if you do have.